So next in line is Vojtova, who is the research group leader of the group of advanced biomaterials, and she will present the latest results of completed and ongoing projects dealing with the issues of personalized medicine.
Okay, thank you, Vojtova. Good morning, everyone. I'm really glad to be here today, to be invited for this sort of workshop. And as Boženka said, I'm going to introduce you our two projects dealing with biomimetic personalized implants for tissue regeneration. Just short introduction of my group. As she said, I'm the leader of Advanced Biomaterials group, and we are focusing on the development of bioactive nanostructured materials for regenerative medicine and tissue engineering, mainly for bone and skin. And we try to make our products that are tailored to the individual patient based on their predicted response or risk of disease. So as Przemek already introduce you, we were involved in actually joint proposal to heal, for example, the bone fusion. So we use biomimetic approach. So we try to emit the tissue, either bone or skin, not only from their structural point of view, but also from their chemistry. So we design and make actually material, in that case, that should replace a disc here in between two vertebral bodies. We designed materials that are from ceramic shell and from polymeric core that can look like that way. And these materials are later tested and implanted. So the project was based on spinal regeneration. The project already ended recently, but it was pretty huge project that many actually partners were involved. As you can see here, it was really nicely multidisciplinary team where there were a lot of surgeries and veterinaries and researchers. And in that case, we really appreciate collaboration with Enantis company that support us with specific proteins in that case. We solve actually the issues with the spine pain. There are a lot of indications for spinal surgery, like degenerative spinal disease, comminuted fractures of vertebra, as you can see here on the picture, spinal malformations, internal disc disruption, bone defects after tumor resection, and other more. And all of these indications are solved by spinal fusion, as Przemek already introduced you, but the fusion is actually based just in between two vertebrae. We actually fuse them together, two segments of the fusion vertebrae. We stop the motion and decrease the pain. So, and how to do that? There are several ways how we can do the fusion surgery. And in the market is actually unbelievably growing with the spinal fusion devices and implants, as you can see here on the graphs. And it's unbelievably growing every year. So there are a lot of money placed already in this sort of issues. So the general gold standard is to use autograft from iliac crest. So, however, this procedure, except many disadvantages, has up to 22% of morbidity, which is, I would say, very terrible. So the modern procedure, as even Przemek said, is to use some sort of cages, metal or PEEK cages. However, in that sort of material, which is not biodegradable and not bioresorbable, there are a lot of non-unions. It means that the fusion actually doesn't happen between these two vertebral bodies. So our innovation is to use a fully resorbable material, which is bioactive. It accelerates healing, it's osteoinductive, and can be personalized for specific patients. requirements. And how we do that, we actually develop this material, and within this project we compare the efficiency of this material with autograft. Here you can see how the autograft was actually removed from iliac crest on the pig animal model, and it was placed in between the spine, in between two vertebrae in spine. Even if the spine is so the autograph was inserted. Here you can see the pig. The pig is not really so happy because of the autograft withdrawing. It's very, very painful. When we implanted only our composite implants, we didn't have to withdraw any autografts. And you can see that the pig actually immediately after the surgery could stand up and could eat and was really pretty much satisfied. And the biggest differences are the results that here, when we use the autograft, you see huge non-unions. So it means that the vertebrae didn't fuse by bone, but with the cartilage. So the cartilage is actually soft tissue, and the fusion was not really perfect. But when we use composite, here you can see really nice bone fusion between two vertebral bodies, which was actually the goal of the whole project. So in that case, the project was very successful, and we are right dealing with some papers and IP. And the other projects actually has a pretty much similar story. However, here we try to imitate skin tissue. We try to imitate two layers of skin, epidermis and dermis. So we designed the materials as the nanofibers on the top, imitating like epidermis, and porous antibacterial sponge on the bottom, imitating the dermis. And the materials looks like that way. And we as well solve this issue within the big project, again supported by Ministry of Health, but here we collaborate with the Burn Center of University Hospital, and again Enantis was actually really helpful in that project that support us with bioactive compounds. We even collaborate with the University of Sheffield and other institutions. And here we solve the issue with the scars of really deeply burned skin. If you can see, the boy was really horribly burned. And after 4 years and 3 resections, you still see that the scars are really horrible, that they are not really nicely healed, because it's multi-step procedure and controlled healing, and the scars are non-elastic. There is actually there is one a product on the market called Matriderm. Is one layer product, but it's really, really expensive, even if it's accepted in Czech Republic. Actually, this part of the implant costs around from 1,000 to 2,000 euros. So it's really unbelievably expensive. So we try to design and prepare new bilateral skin substitute, which can accelerate the wound healing, controls maceration, support elasticity of the skin, and can be personalized. So we develop different kind of materials with different thickness. We test them on cells, even the top nanofibers layer and the bottom layer with different cells to test the cytotoxicity and biocompatibility and safety of the material. And later it was implanted into the defects of the skin when surgeries actually made pretty deep defects in the skin, then remove the whole thickness of the skin and place and inserted our materials there. And we tested the pigs from 1 week to 6 months in the healing, and here you can see our results, that there are healed defects, and in the middle it was controlled group without our material. So you see that they are still missing the dermal part of the skin. There is like hole, pretty deep hole, and no hair, and the scar was not that elastic like the other scars where we used our biomaterial. So in that time, we try to solve the intellectual properties, try to write utility models, maybe even some patents, and we are just talking about offering to the investment these two products right now. So I really thank you for your attention, and I'm happy to answer the question.
I would have just one question before anyone else, because it's really interesting for me. Why pigs as testing animals? Is it because of the bone structure or because of their DNA?
Actually, the pigs are the most similar to human bodies, and it's much better than small animals like rabbits or rats because they heal very fast. And then the pigs doesn't heal that fast. They are pretty much similar the healing process like to human bones or skin. So we can nicely see the differences between the samples. But if we use rat or rabbit, they heal so fast, and then we don't see really any difference because they heal by itself only. So the higher animals or bigger animals are actually much better, and the pigs are really nice models to do that.